And a very warm welcome to our principal ma'am, AGT ma'am, academic administrators, teachers, and all my dear students who have gathered here for today's guest lecture. Let me tell you, Dr. Anis Fatima is from the University of Chicago, and ma'am, we are very pleased to have you here with us. So before we begin anything, let me just present the housekeeping rules that that we expect everybody would follow for the smooth flow of the program. Give me a second before I present my housekeeping rules. Okay, let me just read out the housekeeping rule. Please make sure your mic is on mute during the meeting to facilitate free noise-free environment. Kindly make use of the chat box for quick communication, and the chat box is on the right side of your Google Meet screen. Do not click on the present now button, which is located on the lower right side of the screen, which might create a disturbance. And then the fourth thing that you will have to follow is please turn off your video when the presentation is going on to avoid network issues. When you have to speak, when anybody has a doubt and you have to speak, please unmute and speak one person at a time. And you can turn on your video when you're speaking. Now, now I would like to call upon our AGP ma'am, Dr. Deepti Chaturvedi, for the welcome speech.
Research is to see what everybody else has seen and to think what nobody has thought. Rightly said by Albert George. A very warm and graceful morning wishes to our Honorable Principal Ma'am, enthusiastic students, encouraging parents, teachers, and everyone gathered here. I welcome you all at this guest speaker session. Today we have Dr. Anis Fatima, as Soumya introduced, Senior Compliance Analyst at Northwestern University Chicago. She'll be our guest speaker for today, and she will take us through the lymphatic system. Now, just one line to add over here is that we all know that we are in an age of globalization, and so we are witnessing the sharing of knowledge at two different time zones. This is something when you think on it, it becomes a little bit exciting. No, children? Like you are sitting in the morning and the person who is sharing with you is sitting in the evening. So we are a little confused whether to say good morning to ma'am or say good evening to ma'am. So we'll say good morning, a very warm welcome to Dr. Fatima here from Bangalore. And I hope students, you will enjoy, you will learn, and we will have more such sessions. Over to you, Soumya.
Good morning, everyone. Thank you, Soumya madam and Deepti madam. And thank you, Kala madam, for giving me this opportunity. I'm really excited to teach high school kids. This is my first time teaching high school kids. So please feel free to ask any number of questions you want, and stop me if I'm going too fast, and type chat anytime you want. So today I will be discussing a little bit about lymphatic vascular system, and so I'll be discussing a tiny bit of research that I did in my lab. I mean, the research was really big, the project was really big, but today I'll be discussing very little data. But you're welcome to read my published paper, and you can anytime reach out to me, and I can send you a full text of that paper. We're here to look into a bird's eye view of a lymphatic system. Hopefully, by the end of this lecture, we will get to learn what is lymphatic system and why is it essential for human body. And secondly, we would want to, since I'm a developmental biologist, I thought it is befitting to examine and learn the developmental events that lead to the formation of lymphatic vessels. And this whole process is called lymphangiogenesis. So genesis is formation of new vessels or tissues. And finally, I will be discussing a part of my research that I did to decipher a role of FOXY transcription factors in development of lymphatic system.
Dr. Anise, sorry, I'm very sorry to interrupt in between. Before you start your presentation, I would like to invite our academic administrator, Mrs. Kala Hirimat, for profile reading, for the profile reading. If you can.
Sorry, I think I jumped the gun.
It's okay, no problem. I'll call her upon. Yeah, Kala ma'am, I would like to call you. So now I call Mrs. Kala Hirimat for the profile reading of the guest speaker. Welcome, Kala ma'am.
Kala, I think there's some network issue.
Yeah, it's possible they're traveling, right?
Yeah, she just get down from the bus, so.
So fun fact, me and Kala ma'am went to college together.
Just a little bit of change in sequence. Ma'am, I'm so sorry, but we can continue with your presentation. And Soumya, we will do that profile reading before the interaction session once the presentation is done.
Sure, ma'am, sure.
Sounds great. Thank you, Dipti ma'am. And so basically, these are our learning objectives. And to give a little historical perspective about lymphatic system, Gasparo Asselli, an Italian anatomist in Pavia, is credited for the discovery of lymphatic system back in the 16th century. But due to several technological challenges, like how to image the lymphatic vessels and several other things, and more important things to study, like the cardiovascular system, the lymphatic system research took a backseat. And it actually took a couple of 100 years to actually go forward with research in this exciting field. So lymphatic system is a secondary vascular system. The first one is the blood vascular system, which is very important for all of us. And this lymphatic system comprises of a network of capillaries and vessels. So as we all know, the blood vascular system consists of the heart, the arteries, and the veins. It's a closed loop circular system where oxygenated blood is pumped from the heart and carried through these arteries that are colored red in the schematic, just because they have the blood has RBC cells, the red blood cells. And this oxygenated blood is then carried to tissues where a capillary of a network of arterial capillaries provide the oxygenated blood to the tissues. And once the impure or deoxygenated blood is available, it is carried by the veins back to the heart. So it's kind of a circular loop, and this loop and this circulation happens with the strong pumping action of heart, which is a muscle. So what we are doing here is there is a secondary vascular system, which is not circular. It is basically unidirectional, and it does have a capillary, a network of various capillaries and lymphatic vessels. So this is labeled here in green. So this is basically the lymphatic system. It carries the tissue fluid that is collected between the cells and the tissues back to the heart. So it basically dumps the fluid back in the veins and again back to the heart. So what does this lymphatic system look like? It has a myriad of lymphatic capillaries. These are like very small, thin vessels that basically absorb the fluid. And it has collecting vessels, collecting lymphatic vessels. I'm going to go into a little bit details of each of these components into the presentation. And the lymph nodes. So these are the gateway for immune cells. As you know, immune cells are very important to fight against many pathogens. And the lymphovenous walls. So these are the walls that are present between the lymphatic vessels and the venous vessels. So going into a little more details about how this fluid is produced and how is it taken up in the lymphatic vessels. So if you saw the whole human body, like basically you have a lot of blood vessels. Similar to that, you have a lot of lymphatic vessels. So what happens is when blood flows through artery, there are a lot of macromolecules, proteins, nutrients, and various other solutes in the blood. And the fluid part of the blood is the plasma. And whenever there is pressure increase within the artery, some of the solute some of these components actually try to escape the vessel. So this happens via the pores that are present between the cells. These are endothelial cells that are lining within the artery. So, this might lead to the question, then why don't RBC cells escape? So there is like this size regulation. So RBCs are actually too big to escape the blood vessels. So rest of the important nutrients and things like that, they go into this fluid that accumulates between the tissues and the cells. So this fluid is then taken up by the green vessel here, lymphatic vessel. So this lymphatic vessel is also lined with loosely connected endothelial cells. These are like individual endothelial cells, and they're very loosely connected. And the whole vessel is actually anchored to the tissues by anchoring elements. It's very loosely attached, so loosely attached and takes up this fluid. So this leads to the question. So in the first slide, we did see that heart is the main pump that is that is contracting and relaxing, and that's how pushing the blood in the arteries or like the veins. However, the lymphatic vessels have no pump. So how does the lymph flow through these vessels? So this brings us to the collecting lymphatic vessels. The collecting lymphatic vessels are actually the larger lymphatic vessels, and during development they form later than the capillaries. So these are various capillaries. These capillaries first absorb all the fluid that is present among the tissues, and this then dumps into this huge vessel called the collecting vessel. So these vessels actually are having interspersed walls, like if you can see these V-shaped structures over here, that are walls. So once the fluid is dumped here, the valve opens, and then like so on, like the fluid passes on. So one of the pumping action that comes to these vessels is via smooth muscle cells. So these are muscles actually. And what these smooth muscle cells do is they contract and relax, and this tiny pumping action kind of adds to the pressure that helps the lymph to flow. And another most important push that comes to the flow of lymph is coming from the skeletal muscles. So the skeletal muscles are like your voluntary muscles, like when you want to move a hand or when you want to move a leg or when you're walking. So all these activities actually provide the push to the lymph, and that's how the lymph flows through the vessels. So basically, we saw that we learned that the blood vascular system is a circular system, including heart, the arteries, veins, and there is like strong pumping action from the heart that results in flow of blood in these vessels. So as we just spoke, the lymphatic vessels do not have a specific strong pump for themselves. So how is this lymph actually entering the blood vessels? So when you have like high pressure blood, and then you have this low pressure lymph, how is it possible for the lymph to enter into this deoxygenated blood? So what happens is nature is tricky in that manner. So the lymph takes entry into the veins at a weakest point. That means wherever the blood pressure is the lowest, so almost equal to the lymph pressure, that is the point where the lymph makes entry into the venous system. So in an adult human body, anywhere between 4 to 5 liters of lymph is produced per day and carried every day. So just imagine if all this pool of liquid is just sitting in your body and not going anywhere, how swollen we all will be. So all that activity breaks, especially when you're doing virtual learning, walking and moving your hands and limbs is a great activity to do, not only for your cardiovascular system, but also for your lymphatic system. Finally, coming to the most important, one of the most important component are the lymph nodes. So you have a lot of many lymph nodes in your body. And these are like the gateway to the immune cells. So as you know, immune cells are the ones that are like the fighter cells for your body. They basically fight various pathogens or like the diseases or infections and things like that. So there are these smaller vessels through which the lymph enters the lymph nodes, and a bigger vessel through which the lymph leaves the lymph node. So this is where all the cleansing of the body takes place. Like if there are like dead cells or like cell debris, all these immune cells attack those things, and then a more clear fluid then leaves the lymph node. So lymph nodes are basically good guys, but sometimes they do act like bad guys, especially in the instances of cancer metastasis. So if there is some tumor, and say in liver, and then a few cells dislodge from that tumor, lymph nodes are the one that help propagate those cells and lead to cancer metastasis. So far so good. Anybody has any questions? I'll just keep going. So until very recently, say about 5 years ago, 6 years ago, I'm not very sure about the date. We can always search the date. It was known that the whole of human body is actually having the lymphatic vessels. However, brain is the only organ that is devoid of lymphatic vessels. So this was mainly believed because of the blood-brain barrier. There is a blood-brain barrier in the brain, and that does not allow any fluid or any solutes to cross the barrier, so that brain is protected. However, very recently, in mouse brain, it was discovered that indeed there were lymphatics that were not exactly similar to the rest of the body, but they did do all the functions that our lymphatic vascular system is performing in rest of the body. So here, this is a mouse brain, and these are like the tiny lymphatic vessels. Actually, the surface of the brain is covered by blood vessels. They're called like meninges. And these little tiny green vessels over here in the schematic are the lymphatic vessels. And these are also connected with the lymph nodes in your spinal cord and things like that. So these lymphatic vessels, basically the lymphatic endothelial cells, act in close conjunction with these glial cells that are present in brain. So the glial cells are basically like support cells to a lot of brain functions. function. So scientists have named the lymphatic system in the brain as lymphatic system because it is working with these glial cells. So this brings us to the question that if mouse brain has lymphatic vessels, does human brain has lymphatic vessels? So in a recent study from National Institutes of Health, they showed through MRI imaging that indeed a human brain is, indeed human brain is having lymphatic vessels, and it is definitely doing more of this clearing of solutes, clearing of debris. It definitely looks like very similar to what it's doing in the mouse brain. And also another interesting fact about this lymphatic system in brain is it is active only during awake hours, and it is somewhat dormant during the sleep hours. So, I mean, this is still a very new area where a lot of research is happening. So one of you, or like some of you, when you grow up, maybe you will take up this particular subject and research and find new exciting things. So now that we learned like what the lymphatic system, what is lymphatic system, and it is present in the entire body, it's kind of like super important, just like our circulatory system. So what does this lymphatic system do? Of course, we learned that it is important in like, you know, the immunity, the immune system, and it is also transporting the interstitial fluid back to the blood. And it kind of drains the excess fluid from the tissues. Otherwise, we would all be like swollen. And then it removes cell debris, like basically perform the cleanup action. And last but not least, lymphatic system transports fat from the digestive system. So talking about transportation of fats from the digestive system, in the intestine, the lymphatic vessels have a special name. They're called lacteals. I did not cover a major part of lacteal. That is like a topic for some other day. So what happens if this lymphatic system is disturbed, or like, you know, like having some impairment, or like it's dysfunctioning? So like as I mentioned earlier, it leads to many human diseases. One of them is lymphedema. So edema means swelling. So if the lymph is not transported well, then your body will become swollen. And this is very debilitating in the sense it's very limiting in performing your day-to-day activities. So it's great that our lymph can go where it is supposed to go, and we all can lead normal lives. So another important function of lymphatic system is absorption of lipids, that is the fats. So if the fats are not absorbed properly, then it is like it leads to obesity, and then hypertension. Of course, if the fluid pressure is not balanced very well, leading to hypertension, and an impaired immune response. We all know during this COVID time how important our immunity is to our body, and also inflammation. If there is like, you know, an injury site, and then if the lymphatic system is malfunctioning, it can direct or like misdirect the immune cells, or like the cytokines, towards or away from the site of injury, that thus causing inflammation. So so far, we covered like what is the lymphatic system, and what happens, what are its functions, and what happens when this lymphatic system is impaired or not functioning properly. So as a developmental biologist in the lab, I actually studied the development of lymphatic system. So my question was how these lymphatic vessels are formed. So if they are formed like that, so once we actually know how they are formed, but what are the factors that are influencing the formation of these important vessels? So to go to some of the basics of formation of blood in lymphatic vessels. So when we were all in our mother's womb, like we were like just a ball of cells, right? I mean, it's called blood vessels. So these ball of cells actually, so they have this potency. They're called totipotent in the sense like they have a total potency to acquire any fate. So they can become an eye cell, or they can become a liver cell, they can become a neuron, they can acquire any fate that the circumstances push them to. So some of the, during such development, some of the cells, let's call them as progenitor cells, so they, by a mechanism that is still unknown, they acquire this fate of angioblasts. So angio is like blood, right? So they acquire the precursor fate of angioblasts, which then goes ahead to form a primitive vascular network. So again, here blue are the veins and the red are the arteries. So this whole entire process is called vascular genesis. So it's basically, genesis is like growth of a vascular network, you know. So this is like a primitive network that is formed. So once after this primitive event, a lot of events then lead to formation of mature arterial and venous system. So once the arterial venous cells are differentiated and they have acquired this fate, they're going to be veins for life and they're going to be arteries for life. Some of the cells from the veins then decide to become lymphatic endothelial cells. So these cells then give rise to the whole mature network of lymphatic vessels, which we just studied about. So this brings to some of the research questions that we asked in lab. So our lab was focusing and working on a couple of transcription factors. I'm going to explain a little bit about those transcription factors. So this is the paper we published in Journal of Clinical Investigation. Here we have looked into role of a couple of transcription factors, FOXY1 and FOXY2, in formation of lymphatic vessels. So during lymphangiogenesis, what are these transcription factors doing? So this is like just to show like how questions are asked and like how you can solve it in lab and like gather information. So what are these two transcription factors? First, what are transcription factors? Have you guys studied, do you guys know what are transcription factors? We can type chat. So if we don't know transcription factors, no big. So, so was that a yes or no? Let me see. I want to see my chat box. I actually want to see.
Students, if you have any sort of queries or any questions in your mind, please put in chat box so that we can take it up later on.
All right, sounds great. So, so what happens in transcription? During transcription is like, you know, you have DNA, right? So that's going to be transcribed into RNA. So the transcription factors are facilitating that process. So our lab was studying these two transcription factors, and some of the information we already had on these two transcription factors was they are a member of a four-head box family of transcription factors, and they were actually implicated in cardiovascular diseases. So these two transcription factors actually had overlapping roles. That is like when FOXY1 did something and FOXY2 did that something itself, you know, and whenever they co-localized, they had very similar roles. Actually, we couldn't differentiate like one from the other. So whenever you ask a question or you have a hypothesis in mind, what you do in lab is you hit the literature. You want to know what is already out there, what is already known, and how we can build on our own hypothesis, right? So when we looked into literature, we found that there were like some autosomal dominant FOXY2 mutations that caused lymphedema dystiasis in humans. So this is a disease that is characterized specifically by presence of extra pair of eyelashes. I'm not sure if you are able to see it very clearly, but I can share my presentation with you if you want. You can take a look at it. So these eyelashes actually protrude inward, like you see how our eyelashes are protruding outward. These eyelashes are protruding inward, thereby causing damage to your cornea. The black cornea if you're a black or whatever color you might have. So some of the associated findings in these patients where they had ptosis, that is like, you know how you can bat your eyelid at your will or sometimes not, but these patients had ptosis where they had like drooping eyelid, the eyelid like was just closing on its own, you had no voluntary control of that. And then some congenital heart disease like features were also reported. And cleft palate was seen in some of these patients. So going back to the developmental event slide, we know that the process of by now we know that by process of vascular genesis, a primitive vascular network forms, and then this network of arteries and veins, some of the cells differentiate to give rise to the whole lymphatic network. So, so keeping that in mind, the model in which we studied the development of lymphatic system to check the role of FOXC1 and C2, even if they had any role or no, to test our hypothesis, we studied it in mice. So as you know, the mouse life is a little different from our human life, right? I mean, the gestation period is like about 17 to 18 days. Like when the mouse mom becomes pregnant, they give birth by 17 days. So I'm going to show you some of the timeline of this lymphatic fate acquisition. So in mice, what happens is at E8.5, like 8 days of pregnancy, the embryonic vein is formed, like through the process of vascular genesis. So some of these cells, like by expression of certain genes, here I'm going to mention PROX1, it's another transcription factor. It's like a master regulator of lymphatic fate. So if a liver cell was, if you push a liver cell to express PROX1, it can acquire lymphatic fate. So, so it has that kind of power. So if you have watched Star Wars, it's like Luke Skywalker of lymphatic fate, you know. So at around exactly E9.5 to 10.5, between 9 and 10 days of mouse pregnancy, the venous cells start expressing this particular marker, the PROX1, and acquire the lymphatic fate. And once some of the lymphatic cells are formed through various growth factors, I'm not going to go into details of those growth factors, but these are very specific to lymphatic fate. So it starts forming the jugular lymph sac. So this is the primitive lymph sac, which then gives rise to the whole lymphatic network in mice. So the whole network generation happens around 15 days of pregnancy, and within 2 to 3 days, the mouse give birth to the pups. So for this, the experimental setup we had was we wanted to look into role of FOXC1 and C2, right? So we decided to remove these genes from mice. So we kind of deleted. One model was where we deleted this gene in the entire mouse. So here I'm writing FOXC1 global knockout, but we also analyzed the FOXC2 knockout mice as well. So in these mice, we basically deleted the FOXC1 gene. So there is no FOXC1. So whatever FOXC1 was doing, now there is no FOXC1. So we can see like what functions it was performing, specifically in terms of lymphangiogenesis, is like development of lymphatic system. So the other mouse model that we analyzed was very specific. See, the first one is a global knockout. So we have knocked out FOXC1 in each and every cell of that mouse. So the other mouse that we made was the lymphatic endothelial cell specific knockout. That means we are knocking out these this gene only in the lymphatic cells. So that means we can see the pure function of these transcription factors in development of lymphatic vessels. So I'm not going to make it very data heavy, But I would like to show you some of the very initial results and the problem we saw when we knocked off or when we deleted the FOXC1 and C2 genes. So these are mouse embryos at E15.5, that is 15 days of mouse pregnancy. So when we harvested the embryos, this is the wild type embryo. Wild type means it's the natural one. We haven't tampered with it. We haven't deleted our gene in this mouse. And this is like the global knockout mouse. Here, every cell has no FOXC1. So if you closely observe these two embryos, there is some kind of swelling on this mouse embryo where FOXC1 was knocked off, right? And also there is some swelling on its dorsal side, like in the back. And when under the microscope, when we looked at it, the blood vessels looked very normal. They were like very similar, So, so it confirms that the FOXC1 gene, for some reason, was not having any impact on the formation of blood vessels, but it could be having impact on formation of the lymphatic vessels. So to test our hypothesis, we took a piece of skin from this embryo, the mutant embryo and the wild type embryo, and we stained it to see the blood vessels and lymphatic vessels. And here, the CD31, the red one, is the blood vascular marker. This is the mouse skin. And under the microscope, when you looked at the blood vessels, they look very similar. I mean, they're not thick, nothing is happening, look very like there is a network, and it looks similar between the knockout and the wild type. But when you looked at the lymphatic vessel, live one is the lymphatic marker, the green vessels here, they're kind of thin in the wild type where we have not removed the FOXC1 gene. But if you looked at the mutant where we deleted this FOXC1 gene, you can see how big the lymphatic vessels are, right? I mean, they are like huge and large. I'm sure they are affecting the absorption of lymph, and that's how we see what we see over here. So we went ahead and analyzed the lymphatic endothelial specific knockouts. The results were very similar, very enlarged dysmorphogenesis like vessels in FOXC1 alone, FOXC2 alone, and the double knockout. But what was very interesting is when we removed these two genes, the embryo, some of the embryo had like huge edema over here. You can see like how this mouse embryo looks like as if like there's a lot of water accumulated in this embryo under its skin. So that's how confirming that like somehow the lymphatic system is like not functioning very properly in this embryo. So these embryos actually did not like they couldn't survive. They died in the mother's womb, right? Here we actually harvested, but when we left some of the moms for giving birth, these embryos basically didn't survive. So, so yeah, that's a short story of some of the questions that we asked in our lab. And we published this paper. There's a lot of data. If anybody's interested, you can always check with me. Text. So to summarize, we have hopefully learned that what is lymphatic system, and hopefully we know its functions. And we know that dysfunction of lymphatic system leads to many diseases in humans. And we looked into early developmental events for this lymphatic system. And we briefly learned a little about my research looking into role of FOXC1 and FOXC2 transcription factors in development of lymphatic system. And I did my research at Northwestern University at Dr. Tom Kume's lab. And as you know, nothing is achieved by a single person. So I had a team of scientists supporting my project, and I thank all of them. And we had a lot of collaborators who would provide us with chemicals and things. Everything was a great teamwork. And this whole work was funded by National Institutes of Health. And I'm now open for questions.
Thank you so much, Dr. Anis Fatima. Thank you so much for this wonderful, enlightening latest research that you told us about the lymphatic system. But again, you'll have to excuse me. Before we take in questions, I would first invite our academic administrator, Mrs. Kala Hiramat, for the profile reading. And then simultaneously, students can type in their questions, whatever the questions they have. And later the questions will be taken up by Gauri ma'am. So that's the plan, ma'am.
Okay, sounds good.
Yes, ma'am. So now I would like to call upon Mrs. Kalahi Ramat, our academic administrator, primary, for the profile reading. Kala ma'am.
Yes, I think Kala has lost the connection, so let me do the honors. I'll continue with the profile reading of ma'am because it. Okay, good. Kala is here. Kala, you can take over.
Yes.
She is having continuous network issues. I'm so sorry, Dr. Fatima. So I'll continue with it. Children, I hope you enjoyed the session. It was actually great and informative. Ma'am is a Ph.D. in cell biology from Birla Institute of Science and Technology in 2008. And she completed her MSc in biotechnology from Gulbarga University in 2001. Currently, she is Senior Compliance Analyst in Northwestern University Chicago. Her job profile is like to support the university's conflict of interest programs with an aim to further Northwestern research growth, enforce policy and federal regulations supporting basic science through entrepreneurial efforts at translation. Her research experiences in Ph.D. stem cell biology under L.B. Prasad Eye Institute, Hyderabad, Bits Pilani from Rajasthan. She worked on limbal system cells, developed methods to grow and characterize. Yes, ma'am? You want to add something? Characterize these cells from transplantation in patients with limbal stem cell deficiency. Her postdoctoral fellowship. Children, you all are too small to maybe to understand these degrees, but I must tell you, as on you continue in the process of research, you go for your graduation, then post-graduation, then doctorate comes. After post-graduation, you take a specific research topic and then you go for doctorate, that is your PhD. And once your PhD is done, even there is a higher degree than that, that is postdoctoral. Means you are going one step ahead. And ma'am's postdoctoral fellowship was in the Department of Dermatology, Northwestern University Chicago, where she worked on diversifying role of different receptors and in migration. Research into mechanism controlling these receptors in various cellular functions. She is research assistant professor in Feinberg Research Institute, Northwestern University, understanding the role of factors in development. We understood about that proxy transcriptions in presentation, also development of lymphatic systems. She has got a vast teaching experience and presentations, like in terms of presented papers in North American Vascular Biology Organization, San Francisco. Then in North American, again a paper presentation, North American Vascular Biology, Yale University, New Haven. Then Association for Research in Vision and Ophthalmology. Presented paper on behalf of a fellow scientist at Association for Research in Vision and Ophthalmology. Then in same paper presentations continuously, even in International Symposium of Cell Research Toronto. Then mentored several MD and PhD students, junior postdocs, and associate scientists with academic and research projects. Various awards are there that I can read out for you. Certificate of Appreciation from IMSA that for participation in student inquiry research, guest poster in International Symposium of Human Public Health, New York Stem Cell Foundation Travel Award, Lady Tata Memorial Senior Research Fellowship, India, first prize in confocal imaging contest, that is from Germany. So, what I can sum up is that it is a very, very rich profile, and you should be very happy and proud moment for us to have Ma'am Fatima over here to just enrich us with such research work she shared with us. And children, please come forward with your questions that enrich you more. Over to you. Gauri, you can take over with the questions now.
Yes, ma'am. Good morning, one and all. Dear participants, the session is open for discussion. So we have a few questions in the chat box. So first is from Navneet Suraj from grade 9. So Navneet, you can just unmute your audio and ask your questions directly to ma'am.
Yes, ma'am. Ma'am, are there any methods for curing the dysfunction of the lymphatic system, be it in the form of vaccines, antibiotics, or drugs? And if so, is it a lifetime medication or is it a one-time cure?
Good question, Navneet. So depending on where you are, right, depending on what extent of disease you're facing, there are different ways of managing it. In humans, especially when like there is a congenital form of lymphatic dysfunction where like on a specific side, the babies get this swelling. So that is actually managed like by a one-time surgical management. But if people are like say, for example, in terms of cancer metastasis, like when cancer has spread throughout your body, you'll have to remove lymph nodes. That leads to a big lymphatic problem. So that's kind of a debilitating disease that you have to live with your life. So it is managed like diabetes, it is managed, but it can never go away. Does it answer your question?
Yes, ma'am.
Thank you for asking.
Thank you, ma'am.
Thank you, ma'am. Thank you for the clarification. So we have one more question from Arvind Kottamangala from grade 9. So Arvind, go ahead.
Ma'am, I recall that you said that transcription factors convert DNA to RNA. Why is that necessary?
So that's a good question too. So basically, you know, in DNA you have all these genes, right? And these genes need to be like translated into like all the genetic information is encoded in the DNA. So whatever your body needs, this needs to be translated in terms of messages. Either they are proteins and things like that, right? So there are 3 steps to converting this genetic message. So one is transcription, where DNA is changed to RNA, and the RNA is then further translated to proteins that can perform the function for your whole body. So you will study these various like biological phenomenon in detail, but that's the like short nutshell answer. Like there are 3 steps for anything that needs to function in your body, like the message is converted from DNA to RNA and to RNA to the protein.
Okay, ma'am. Thank you.
Thank you, ma'am. We have one more question from Ruchira from grade 10. Please, Ruchira.
Thank you, ma'am. Ma'am, during the presentation, you told us that there's another disease that's the lymphatic dystasis, due to which there are extra eyelashes. Does that cause blindness in people?
Yes, very interesting question, Ruchi. And it does cause blindness because, you don't want anything to like scrub your cornea, so that the black cornea or like blue cornea if you have blue eyes. So you don't want anything to disturb the surface. So that's why eye is so protected. Like you bat eyelids all the time, just because you don't want the debris, anything that hurting your eye, right? This is eye's like safety mechanism. If something flies in your eye, the eyelid shuts by itself, right? It's a safety mechanism. Evolutionarily, we have developed that mechanism to protect your eye. So if this eyelashes are like just protruding inside, they will disturb the cornea, and then that could eventually lead to blindness. So, yeah.
Thank you, ma'am. Ma'am, I have one question. So as this FOXC1 and FOXC2 genes, which codes for transcription factors, so that deletion can be only congenital or it can be acquired one?
So so this is like we are doing it in lab, right? So what we did was for us studying the timing of this lymphatic development was very important. The time window from E10.5 to E15.5, right? So we actually, the way we designed our mouse was we could control the time of when we want to delete it.
Okay, can stem cell therapy could help in this process?
So, so you're asking about the autosomal dominant mutation in humans?
Yes, ma'am.
So, yes and no, I mean, we are very far from getting cured for that. I mean, I'm not going to never say never, right? So things might come up, and like among all these bright students that are here, they might actually discover something related to this. However, as far as I know, nothing of that sort is happening currently.
Okay. Thank you, ma'am. Thank you so much. Over to Soumya.
Thank you, ma'am. Thank you, Gauri, ma'am, for taking up the question answer session. And thank you, Dr. Anis, for clarifying all the doubts. And I would like to personally thank you, since being a microbiology student and listening to you, and Gauri ma'am was asking you questions being a biochemistry student. And also thanks on the behalf of students whose questions have been answered. Thank you so much for telling us about the blood vascular system and the lymphatic system, which not many people know about. And owing to the current corona pandemic, I guess it's very much necessary that people know about it. Yeah. So now I would like to call upon Edwin, our head boy, for vote of thanks. But before that, I would request all the students. Deepti ma'am has shared a feedback link on your in your chat line. So kindly give your feedback before students exit. Now I request Edwin to take over for the vote of thanks.
Before that, thank you, Soumya ma'am and Gauri ma'am, is it? So it's always happy to meet fellow biologists, So it's speak the same language, right?
Yeah.
It was actually a great experience from all our side, ma'am. It was actually a great experience for all of us.
That's awesome. I'm happy about it.
Edwin, you can take over.
A graceful and warm morning to our most valued guest, Dr. Anis Fathima Khan, enthusiastic teachers of Edify, and my dear friends. It is my privilege to propose a vote of thanks on this occasion. I, Edwin Jason, the head boy, on behalf of Edify School, first of all, extend my most sincere thanks to our honorable guest, Dr. Anis Fatima, for her knowledgeable session. A big thank you to our beloved principal ma'am, Ms. Pamela Ghosh, for her support. I also take this opportunity to thank Ms. Aditi Chaturvedi ma'am, the AGP, for her warm welcome speech. Last but not the least, I would also wonderful audience for the session. Thank you, everyone. Have a wonderful day ahead.
Thank you, Edwin, for vote of thanks. Students, please fill the forms, the feedback forms, and I guess Dipti ma'am, now they can exit the session, right?
Yes, the students can fill the feedback form and then they can start exiting. And ma'am, I hope Kala is having your mail ID, so even if we want to get in touch with you and if any student, so if we can post situations that way or some feedbacks we can share with you. So can we have your mail ID? Not here.
Yeah. Yeah, no,
Yeah.
Kala has my email ID, and I know the students have asked for the full-length paper, the data that I just presented. I'm going to share the paper with Kala ma'am,
Yes.
and
Few students are
I'll
interested.
be,
He has said that he is interested to see the paper. Right,
yeah,
ma'am.
yeah.
Right. So we will be getting in touch with you again with that, the
Yes.
feedback and the students' reactions and their questions, of course, because this is the best way out to enrich them and they'll get to know something they were not aware of earlier. So it is going to be great for them also as well.
This is like a great initiative on behalf of your school, actually. I'm very happy. I mean, I think it is all our duty to bring science to the schools. So if the student can learn questioning early, they can go really far. So, I mean, these are scientists, right? Yeah.
Yes, ma'am. Thank you. Thank you for being so friendly and so easy for us to reach you out in that way, because even I'm sure the students must be feeling the same way, that it was like very informal and friendly interaction. They felt that we felt in the same way. It was really nice because for the very first time when we interact and we interact in a very informal way, it becomes easy to learn actually.
I agree. I agree. The goal is to learn.
Yes, definitely.
Thank
Thank
you,
you, ma'am.
everyone. Have a great day.
Thank you. Thank you.